Hi everybody, and welcome to our module on apoptosis. Apoptosis is also called programmed cell death, and it gets its name because the cell uses its own machinery, its own programming, to destroy itself. The cell activates its own enzymes, and this leads to cell death. In contrast to necrosis, apoptosis is a very orderly process. The cell membranes remain intact, inflammation is not activated, and ultimately phagocytes will consume the cell that has died. A single cell can undergo apoptosis without disturbing any of its neighbors, and this is in sharp contrast to necrosis. In necrosis, the cell basically falls apart. This triggers inflammation, and often multiple cells die at the same time. That is not true for apoptosis, and for that reason, this is the preferred way for cells to die in the body because it's neat and orderly and doesn't destroy any other tissue. Apoptosis is an active process. It is ATP dependent. It requires an energy investment by the cell. And this is in contrast with necrosis, which does not require energy. And the idea here is that in apoptosis, the cell uses its energy so it can die in an orderly way and preserve the overall organism. In contrast, necrosis is where a cell dies in a disorderly way. This leads to inflammation and often damages surrounding tissue. And we're learning more and more that many stimuli that we used to think cause only necrosis actually may cause both apoptosis and necrosis. For example, myocardial ischemia was historically thought to lead to necrosis, and it does, but there's now evidence that it actually leads to both forms of cell death, apoptosis and necrosis. What it looks like happens is that initially when there is ischemia, cells undergo apoptosis. This lets some cells die in an orderly way and avoids inflammation and necrosis. However, later those myocytes run out of ATP and they can't die by apoptosis because that requires ATP. At this point, the cell becomes damaged and undergoes necrosis. And there's more and more evidence that apoptosis and necrosis are linked in this way and that the body tries to have cells undergo apoptosis first and only proceeds to necrosis when apoptosis fails, often from depletion of energy. Let's talk about some causes of apoptosis. Why would a cell undergo apoptosis? Well, first of all, apoptosis is a normal part of embryogenesis. Many cells develop in the embryo but do not persist into the newborn baby and adulthood. This is part of the normal development of the fetus. When hormones are withdrawn, many tissues undergo apoptosis. There are many so-called hormone-dependent tissues, and when you withdraw the hormone, those cells die by apoptosis, which is much better than dying by necrosis, which would cause inflammation. So, for example, the endometrium can grow when stimulated by progesterone, but when that is withdrawn as part of the endometrial cycle, apoptosis occurs. And it's good that apoptosis occurs because if it didn't and necrosis happened, you would have an inflamed uterus every time the menstrual period occurred. Immune cells often undergo apoptosis. T cells in the thymus do this, so do B cells in the germinal centers of lymph nodes. In the thymus, cells that react with self-antigens, T cells, will undergo apoptosis so that they won't survive to attack the body itself. In germinal centers, when you have inflammation, B cells multiply, but when the immune stimulus is resolved, those B cells undergo apoptosis, so they die in an orderly manner without causing the lymph node to become inflamed from necrosis. When DNA damage is detected inside of cells, those cells will often undergo apoptosis. Cells that are abnormal in any way, their machinery is off, their proteins are off, this is often detected, and the cell undergoes apoptosis. And then finally, many infected cells, especially those infected with a virus, will undergo apoptosis. We'll talk later about how CD8 T cells can actually trigger this process to occur. So there's lots of basic science literature on how apoptosis occurs. The reason this has been so heavily studied is because many cancers develop from failure of apoptosis. A cell that is abnormal should undergo programmed cell death, but this doesn't occur, and the cell is allowed to grow and multiply and ultimately becomes a tumor. So let's talk about some of the current understanding of the mechanisms of apoptosis. The first thing to know is that enzymes called caspases are critical to apoptosis. These enzymes are found in the cytosol in an inactive state, but when they become activated by a variety of triggers, this can lead to apoptosis. There are two pathways that have been described that lead to caspase activation. The first one is called the intrinsic or mitochondrial pathway, and this gets its name because this pathway is initiated by the mitochondria, which is inside or intrinsic to the cell. The second pathway is called the extrinsic or death receptor pathway, and this gets its name because this is activated by things outside or extrinsic to the cell, and it happens because of interaction with death receptors found on the cell membrane. Let's talk about the intrinsic pathway first. This pathway begins with opening of pores in the mitochondrial membranes. And when they open, they allow cytochrome C to release into the cytoplasm. By way of review, here's a slide from the biochemistry video on electron transport, and cytochrome C participates in electron transport by transporting electrons from complex 3 to complex 4. It's also used as a trigger for apoptosis when cytochrome C is released into the cytosol. What cytochrome C does is it binds to something known as APAF1 or apoptosis activating factor 1, and this can activate caspase enzymes. The ability of cytochrome C to get into the cytoplasm and activate caspases is controlled by a family of proteins called BCL2 proteins. Some of these are pro-apoptotic, meaning they favor apoptosis, and some are anti-apoptotic. And the balance of activity determines whether a cell undergoes apoptosis. So if the pro-apoptosis proteins are more active, then the cell will undergo apoptosis. If the anti-proteins are more active, then the cell will not undergo apoptosis. There are many proteins that have been described that are part of the BCL2 family. Some that have been described that are anti-apoptosis include BCL2, BCLXL, and MCL1. These are found in the membranes of the mitochondria, and they basically keep the membrane intact and prevent cytochrome C from leaking out into the cytosol, where it can go on to activate caspases. BCL2 family proteins that are pro-apoptosis include BAX and BAC. These are the two famous ones. They bind to these anti-apoptosis proteins and stop them from doing their job. This opens pores in the mitochondrial membrane and allows cytochrome C to leak out. Therefore, BAX and BAC promote apoptosis. And an easy way to remember this is that the BCL2 proteins with an A in their name are for apoptosis. They cause apoptosis. That's BAX and BAC. Those are the ones with the A in their name. All of these anti-apoptosis proteins do not have an A in their name. And here's my own little drawing of how the intrinsic pathway works, which I show you to help you remember it. Keep in mind, however, that the actual intrinsic pathway is far more complicated than this. This is an oversimplification, just so you can remember the details for your boards. So under normal circumstances, cytochrome C is found within the mitochondria, and BCL2 and other anti-apoptotic proteins are keeping it inside the mitochondrial membrane. If a trigger activates BAX or BAC, they can move the anti-apoptotic proteins out of the way. This allows cytochrome C to come out into the cytosol. It interacts with APAF1 and activates caspases. A very high-yield clinical example of the intrinsic pathway gone awry is follicular lymphoma. This is a subtype of non-Hodgkin lymphoma that I discuss in the hematology section. It's a B-cell malignancy. And this malignancy has been shown to overexpress BCL2. And remember what I told you, BCL2 is anti-apoptosis. So when there's overexpression of this protein, those mitochondrial pores will never open. This means caspases cannot be activated and the cells will never undergo apoptosis. That leads to uncontrolled cell growth. And as I told you before, many malignancies are believed to derive from a failure of apoptosis, and this is an example of one that has been specifically tied to one of the BCL proteins. Let's talk about some of the triggers for the intrinsic pathway of apoptosis. If a growth factor is withdrawn, cells will often undergo the intrinsic pathway and undergo apoptosis. So in hormone-sensitive tissues like the uterus, if the hormones are withdrawn, that will lead to cells undergoing apoptosis by the intrinsic pathway. Cytokines drive the growth of immune cells, but when those cytokines go away, those immune cells will undergo apoptosis by the intrinsic pathway. And also the intrinsic pathway is heavily involved in embryogenesis. When DNA damage occurs, either from radiation or there are many chemotherapy agents that damage DNA, this can lead to activation of BAC and BAX, which are pro-apoptosis. The protein P53, which I talk about in other videos, is an activator of BAC and BAX. And then finally, if there are abnormal proteins inside a cell, this can happen when there's heat exposure or hypoxia or low glucose. This can often trigger apoptosis via the intrinsic pathway. Now let's talk about the extrinsic pathway of apoptosis. This pathway involves death receptors. These are found on the surface of cells, and when they're activated, they lead to apoptosis. All of the death receptors are part of a family of receptors called the TNF family, or the tumor necrosis factor family, and they span the plasma membrane and spread into the cytoplasm from the outside of the cell. One of the best described death receptors is called FAS. It's also called CD95. It can bind to the FAST ligand, and when this happens, this triggers a sequence that leads to the activation of caspases and therefore apoptosis via the extrinsic pathway. So let's go through some examples of apoptosis via the extrinsic pathway. One of the best examples is when a CD8 positive T cell kills a virally infected cell. and I talk about this in more detail in videos in the immunology section. When a cell is infected with a virus, that cell can present viral antigens on the cell surface via MHC class 1. MHC class 1 can bind to CD8 T cells, and when this happens, the CD8 cells will release substances called perforin and granzymes into the virally infected cell. One of the first things that can happen is these perforins and granzymes can directly activate caspases, leading to apoptosis. But in addition, CD8 T cells produce the Fas ligand, and that can bind to Fas or CD95 on the surface of virally infected cells, and this also can activate the extrinsic pathway apoptosis so that the cell infected with a virus will die. Another example of the extrinsic pathway is the thymic medulla, and we mentioned this earlier. T cells that bind to self-antigens in the thymus will die. This is called negative selection, which I talk about in videos in the immunology section. And when T cells bind self-antigens, death will occur via apoptosis via the extrinsic pathway, and this has to do with Fas ligand interactions. This interaction occurs between T cells and thymus cells, and that leads to apoptosis. There is a rare disorder called autoimmune lymphoproliferative syndrome, and the reason you should know about this for step 1 is because the underlying cause is a defective Fas ligand pathway for apoptosis. In this disorder, Fas and Fas ligand do not normally bind to each other and trigger apoptosis. For this reason, there is poor negative selection in the thymus. This means that many T cells that bind self-antigens will survive. This leads to an overproduction of lymphocytes, and you have lots of T cells around that can react to some degree with self-antigens. So this can lead to lymphadenopathy, hepatomegaly, and splenomegaly. This is all due to lymphocytes in these organs and structures that are growing by reacting with self-antigens. There's a very high risk of developing lymphoma, and there are many autoimmune diseases. The details of this disorder are not important, but you should understand that it is caused by a defective interaction between Fas and Fas ligand and an ineffective extrinsic pathway occurring among T cells in the thymus. So now let's talk about some of the cellular changes you can see under the microscope if you watch cells as they undergo apoptosis. First of all, cells undergoing apoptosis will shrink, and this is in contrast to necrosis. Cells undergoing cell death via necrosis always swell. Second classic change is chromatin condensation in the nucleus. This is also called gnosis. You will see the nucleus condensing. This is shown on this slide on the screen here. Parts of the nucleus clump together and become dark, and this is one of the hallmarks of apoptosis. The membranes of cells undergoing apoptosis will form blebs. This is also seen in necrosis. So if we look at my slide on the screen here, the top is a normal cell. In the middle panel, the cell is undergoing apoptosis. So you can see that the nucleus is condensing and these blebs are beginning to form in the membrane. The cell is shrinking as well. And then finally, what the cell does is it basically explodes and releases these small little packages of parts of the cell. These are called apoptotic bodies. They're fragments of the membrane that contain cellular organelles, and they contain ligands for phagocyte receptors. So phagocytes will recognize apoptotic bodies and consume them. And this is shown in the bottom slide. The cell breaks apart, forming these little packages, the apoptotic bodies, and they get eaten up by the phagocytes. This is a nice orderly process of cell death. It does not cause inflammation, and it's the preferred way of the body to have cells undergo cell death. I'll briefly mention DNA laddering. This is a technique used in the laboratory to identify that cells have undergone apoptosis. One of the things that apoptotic caspases can do is to cleave DNA, and when they do so, they cleave it at very specific regions, and they always form pieces that are multiples of 180 to 185 kilobase pairs. So for example, one piece might be 180 kilobase pairs, another piece might be 2 times 180 kilobase pairs, and another piece might be 3, and so on. But they're always multiples of 180 to 185. When you spread these out on a gel, you form a ladder of sizes as shown in the bottom right side of the screen. And when you see this nice orderly ladder where the pieces are all multiples of 180, you know that apoptosis has occurred. In contrast, cells that undergo necrosis have random fracturing of the DNA, and this leaves pieces of DNA that are multiple different fragment lengths in size with no association to one another. I'll finish with this slide highlighting some of the differences between apoptosis and necrosis. Apoptosis is non-inflammatory, whereas necrosis evokes an inflammatory response that can damage neighboring cells and tissues. Cells shrink in apoptosis, they swell in necrosis. Membrane blebbing is common to both modes of cell death. The membrane remains intact in apoptosis as apoptotic bodies are formed. The membrane is damaged and contents of the cell leak out in necrosis. A single cell can be affected by apoptosis without disturbing its neighbors. In contrast, necrosis rarely happens to a single cell. It usually happens to tissue beds, and the inflammation of necrosis draws in other cells to the area of damage. DNA laddering is seen in apoptosis. It's not seen in necrosis. And then finally, apoptosis can be physiologic. For example, it can be part of embryogenesis or part of the menstrual cycle. However, necrosis is always pathological. It always means some pathologic disorder is occurring. And that concludes our video on apoptosis.